Okay, next up is Amy Jaramillo-Lambert, and she's going to tell us about a unique role for topoisomerase during male meiosis.
Yeah, I couldn't see over the podium. Okay, first I'd like to thank the organizers for allowing me to tell my story today. So my research focuses on the specialized cell division of meiosis, which allows diploid organisms to generate haploid gametes for sexual reproduction. And during meiosis, chromosomes first replicate their DNA, and then homologous chromosomes must pair, synapse, and form physical connections through crossover recombination, which is essential for the segregation of the homologs at the first meiotic division. And this is followed by a second meiotic division with no intervening S phase, resulting in 4 haploid products. And to ensure the proper execution of all these events, it takes a complex interplay of many proteins. And today I'm going to tell you a story about one of these proteins called TOP2, specifically asking how does TOP2 promote faithful segregation of chromosomes during male meiosis. So TOP2 encodes the C. elegans homolog of topoisomerase 2. These topoisomerases solve topological problems such as DNA catenase or DNA supercoils by creating transient breaks in the DNA. These enzymes are conserved from bacteria to humans. They've been well studied in mitosis, where it's been shown to play a role in solving topological problems in DNA replication, transcription, recombination, sister chromatid separation, and chromosome condensation. And because of its many roles in mitosis, it's an important cancer chemotherapy target. However, these chemotherapies are highly toxic, and one of the side effects is both male and female infertility, suggesting that it also plays roles in meiosis. So there has been a few studies that have looked at topoisomerase 2 in meiosis, and it's been shown to be required for segregation of homologous chromosomes at the first meiotic division. But its mechanism of action during meiosis is unknown. So we identified a new allele of topoisomerase 2 called TOP2IT7. It's a temperature-sensitive allele. At 15 degrees, these animals are wild type, but if we shift them to 24 degrees, this causes embryonic lethality. And we found that the embryonic lethality in hermaphrodites could be rescued by mating to wild-type males, suggesting that it's a sperm defect. So to test this, we mated mutant males raised at 24 degrees with wild-type females and asked, does this result in dead embryos? So this graph is showing the average percent embryonic viability at both 15 and 24 degrees. So wild-type males that have been raised at the permissive temperature of 15 degrees mated to wild-type females have high levels of embryonic viability, and this is also true at 24 degrees. TOP2IT7 males that have been raised at 15 degrees mated to wild-type females also have high levels of embryonic viability, but if we now raise these animals at 24 degrees and mate them with wild-type females, this results in dead embryos, showing that it is indeed a paternal effect embryonic lethal allele. Because this phenotype resulted in embryonic lethality, we started characterizing this mutant by looking at the early events of embryogenesis. And in these images, the DNA or histones are tagged with M-cherry, so the DNA or chromatin is in red. There's also NPP1 GFP, so the nuclear envelopes are in green, and tubulin GFP, so we can visualize both the meiotic and mitotic spindles. So in wild-type animals, shortly after fertilization, the paternal DNA is this compact structure of chromatin in the posterior of the embryo, and it hangs out there while the maternal DNA completes the meiotic divisions. After meiosis is complete, both the maternal and paternal DNA undergo a chromatin rearrangement, and they form pronuclei, and at this stage you can see the centrosomes that are brought in with the sperm. The maternal and paternal pronuclei will migrate and meet up towards the center of the embryo, where they'll begin to set up the first mitotic spindle. In IT7 animals, after fertilization, we can see the maternal DNA undergoing the meiotic divisions, but we didn't see any paternal DNA, and this becomes more apparent after pronucleus formation, because we can see the maternal pronucleus and the centrosomes that are brought in by the sperm, but no male pronucleus. And even in the absence of paternal DNA, the maternal pronucleus will migrate and meet up with the sperm-provided centrosomes and start to set up the first mitotic spindle. And this is just to show that in 100% of the embryos examined, they all had a lack of paternal DNA. So because these animals didn't have any paternal DNA, we wanted to examine the male germlines to find out when and why these males or these embryos lack paternal DNA. So this is a cartoon of the male germline. The germline is in a syncytium and arranged in a spatial temporal order along the distal to proximal axis. The distalmost end of the gonad has a population of mitotically dividing cells. As these germ cells progress down the gonad term, they enter meiosis. They'll undergo an extended meiotic prophase and do the meiotic divisions in the proximal gonad. So the next slide I will be showing you will be focusing on the meiotic divisions. So just for anybody who may not be familiar with male meiosis in C. elegans, and just a blow up of this region, the germ cells will first form primary spermatocytes. They'll set up, the chromosomes will align along the metaphase 1 plate. Then they'll undergo the first meiotic division, and this can be followed by either an incomplete or complete cytokinesis. They'll form secondary spermatocytes. They'll arrange the chromosomes on the metaphase 2 plate, undergo the second meiotic division, and then they'll undergo spermatid budding, where the haploid complements of DNA are segregated into the spermatid buds, and all non-essential components are dumped in the residual body for removal. So we wanted to examine the meiotic divisions, and we did this by HUXT and DIC imaging. In wild-type animals, we can find primary spermatocytes with the chromosomes arranged on the nice metaphase plate. They'll undergo the first meiotic division, the anaphase, form secondary spermatocytes. They'll set up the metaphase 2 plate, undergo the second meiotic division, and form spermatid buds. And you can see the DNA segregated into the spermatid buds. In TOP2 animals, we can find primary spermatocytes with the chromosomes arranged on the metaphase 1 plate. They appear to try to at least attempt the first meiotic division. However, the chromosome morphology is highly abnormal. I never observed any secondary spermatocytes, but they do seem to get from this first attempt at division to the spermatid budding phase. But you can see here that the chromosome morphology is highly abnormal, and these two chromatin masses appear to be connected by a chromatin bridge. And the chromatin is trapped in the residual body and fails to segregate to the spermatid buds, resulting in the anucleate sperm. So topoisomerases have known roles in DNA replication, and the male germline, or C. elegans germlines, have both, or sorry, never mind. So the next question I want to ask is, do the meiotic chromosome segregation defects of TOP2 stem from defects that occurred during premeiotic S phase? So this is an image of a male germline that's been dissected out of the worm and stained with DAPI. So premeiotic DNA replication occurs in the distal germline just prior to entry into meiosis. And we know that it takes about 20 to 24 hours for germ cells to move from the distal gonad, progress through meiotic prophase, and enter the meiotic divisions. So we wanted to look specifically at the meiotic divisions, so we decided to do some short temperature shifts. So if we shift germs or males to 24 degrees for 4 hours, and then examine the germ cells that are undergoing meiosis, these are germ cells that were only in mid to late packaging at the start of the temperature shift and would have completed premeiotic DNA replication at the permissive temperature of 15 degrees. So now we're only assessing the effect of mutant TOP2 during meiosis. And the image I'll show you will be focusing on the proximal gonad. So in wild-type animals at 24 degrees for 4 hours, we can see the compact chromatin structures of the condensation zone and the post-meiotic DNA, and then we can also find examples of germ cells that are undergoing the meiotic divisions, and here's 2 metaphases and an anaphase. In TOP2 animals that have been shifted to 24 degrees, we can also find the compact structures of the condensation zone and some post-meiotic sperm DNA, but we still observe these chromosome segregation defects, indicating that these were germ cells that underwent premeiotic DNA replication at 15 degrees, and so these are defects that are not due to defects that occurred in S phase. So given that type 2 topoisomerases solve topological problems that arise in DNA, we next asked if TOP2 was playing a role in solving topological problems that might arise in crossover recombination. So So to test this hypothesis, we made double mutants of SPO11 and TOP2-IT7. So SPO11 is the enzyme that creates the double-stranded breaks to initiate meiotic recombination. So in the controls, the TOP2 homozygous animals with SPO11 heterozygotes, there's no chromosome segregation defects at the permissive temperature of 15 degrees. But if we shift these to 24 degrees, we see these abnormal chromosome morphologies indicative of chromosome segregation defects due to the mutant TOP2. In the SPO11 TOP2 double mutants, at 15 degrees, we also don't observe any chromosome segregation defects, but these double mutants raised at shifted to 24 degrees, we still observe chromosome segregation defects, suggesting that removal of crossover recombination does not ameliorate the chromosome segregation defects of TOP2. So in conclusion, I showed you today that we identified a new allele of topoisomerase II called IT7. It's a temperature-sensitive allele that causes mutant hermaphrodites to lay dead embryos at 24 degrees. And this is due to a meiotic chromosome segregation defect during spermatogenesis that results in anucleate sperm. And we also found that chromosome segregation defects can arise independently of premeiotic DNA replication and meiotic recombination. So right now, kind of given the genetic characterization of this mutant, and we kind of favor a different hypothesis. So we know that these problems can arise independently of DNA replication and recombination, which are well-known roles for topoisomerase II during meiosis. So now we favor the hypothesis that maybe topoisomerase II is playing a role in the chromosome conformation, specifically in the male germline, and that's contributing to the sex-specific differences that we see between the hermaphrodite and males. And so now we're working on characterizing these genetic interactions. So I'd like to thank the members of my lab, my PI, Andy Golden, and past and current members that have contributed to this project. Other labs at NIH for their helpful comments on this project, and Harold Smith for the identification of IT7 as topoisomerase II. And all of you for listening today. Thank you.
Questions?
So why do you think TOP2 only function in sperm but not in oocyte? Do you think that there are it's temporally controlled, so that's only required during the first part, and maybe in hermaphrodite there's something else that's redundant, or what's your hypothesis?
So, right, so our current hypothesis, that kind of what we think, so in the male germline, in the proximal gonad, the males undergo this condensation zone where the chromosomes will aggregate into this mass called a karyosome, and this does not occur in oogenesis. And that's kind of where we think that topoisomerase II might be playing a role.
Hi, Amy. That was lovely. I was just wondering, this is a bit crazy question. So one thing that struck me was that even when in meiotic mutants in the sperm, the chromosomes don't segregate and are misbehaving, then they can pull out into the spermatids some DNA because the centrosome just does all that work. Did you look at the centrosomes in the top one in this budding spermatids?
Did I ever get the centrosomes in the spermatids?
Yeah.
I never saw centrosomes in this.
You see it in embryos.
Huh? I guess I shouldn't say, I haven't specifically looked at that. The centrosomes they look okay when I look at tubulin staining. I mean, like, I don't see like multiple centrosomes or any in the divisions. So
because I saw that you have doing live imaging. So when you're looking at the, as the second meiotic division is going on, it looks like they're not just all sitting in it's like they're trying to attach or connect to the spindles, but they're not making it to the budding spermatid, right?
Right.
do you feel that they're attached and then they're trying to make it to the spermatid, or they never even try?
So well, yeah, I think that they're trying, but they can't make it.
Okay, great, thanks.
You're welcome.
Nice talk. I was wondering, because the role of topoisomerase II is in resolving concatenation, is there a way to look at any kind of breakage during meiotic divisions that may be evident because that isn't happening? Because that could be, the chromosome segregation defects you could be seeing could be caused because those strands of DNA aren't getting resolved.
Are you talking about breakage and recombination, or like topoisomerase's specific breakage?
No, I'm talking about in the meiotic divisions when the chromosomes don't segregate properly and you see these bridging events.
Yeah, I
that
haven't.
The concatenation resolution that should be happening then to pull apart the chromosomes, potentially, is there evidence that maybe thinking along those lines of how that could be happening or failing?
Yeah, so I haven't looked. I don't. So based on where we know this mutation occurs, so we don't think it's probably not even interacting with the DNA. So I don't think it's like getting there and then being like I don't think it's doing its enzymatic activity because it's not associated with the DNA.
Thank you, Amy.